All right, I thought we would finish up our discussion of gene interactions by working some problems, some epistatic practice problems. Now, epistasis, as you can see, we talk about it in a generally a two locus system where we have A and B, loci, and then alleles under each, but really can be 3, 4, 5, 6. I mean, in the real world, epistasis is a big part of our genetic makeup and how we produce these phenotypes. But what I'd like to do is to just do a few problems and kind of show you, okay, these look horribly complex when you first see them, and they certainly would be the ways you might see them on exams. But in reality, they're not that difficult. In fact, if we think about it, if we just use Mendel's Big Six, that's all we need. That's all we need to work these problems and to know a few ratios. But other than that, in fact, some of these problems aren't even going to have ratios. We won't even worry about it, okay? But I would get my Mendel's Big Six out and let's take a look at some of these problems. All right, so here's one typical problem. Okay, in this one we've got some cats, so we've got some mammals. We're told the mode of inheritance is a dominant epistatic allele. Okay, so it's a dominant epistatic. W produces white. In order to have color, apparently you need to be little w, the recessive condition. So this is the light switch, right? On, off, on, off. This time the light switch is dominant, which is off, right? Big W. And you have to see the lights, you have to have two switches basically turned on. Now, once you get the light turned on, then you can either be black or orange, okay? And this is a codominant sex linked. Oh my gosh, now we got epistasis, codominant, sex linkage all in one problem. Oh, okay, let's not worry about that. Let's just think about how you work problems in general, okay? So what we've got, it says homozygous females are either orange or black. The heterozygote is called tortoiseshell or maybe calico. Tortoiseshell is usually the more common name we see in genetics, okay? So what do we got? We got a two-allele system. Either you are homozygous orange, homozygous black, or you're a het and you're tortoiseshell, okay? So these are cats to show you what it looks like. That's an orange cat, obviously. Here's a black cat. And these two at the bottom are tortoiseshell. And tortoiseshell just depends, they can be blotchy, very blotchy all over, or they can have just big blotches like this. Now, just to kind of remind you, and I've seen this in the past, but just in case you haven't in a while, I'll remind you how this happens. It happens because in mammals, okay, all mammals, we have, of course, XX and XY. We have the ligase mode of sex determination. We get two X's if we are female. We get one X and this Y if we are male. Okay. Well, if you're female with these two X's, we don't need them. Okay. It turns out. So during fetal development, fairly early on in fetal development, one of those is put away. Okay, and it forms what's known as a Barr body. It was discovered by Murray Barr in the 1950s, so it uses their name. But this is a cell, a female mammal cell. Okay, we could take a cheek cell out of any of you and look to see whether you had these or not, depending on your sex. If you're a female, you're gonna have these dark staining little spots along the side of the nucleus. So this is the nuclear region. That's the DNA right there, and this big cell around it. These are called Barr bodies. They're simply one of those chromosomes that gets packed up by condensation, okay, highly packed up, all wrapped up in nucleosomes and just put over out of the way because we don't need it. We only use one X, okay? Males, of course, don't have those, right? Forms. Now, what this does is because it's sex linked, Mary Lyons proposed in the 1960s that it was random. Of course, we get one X from dad, one X from mom, and it was either your dad's X got used or your mom's X got used, okay? And it depends on which one of those. It's just a random event. Now, it turns out it's not particularly random. There are some things that go on, but for our purposes, we're going to call it random. Certainly in skin cells, okay, things that are epidermal cells, it pretty much is random, and that's why you get these calico cats, because they have places where the cells are starting to produce, okay, they form out the epidermal cells, and then they shut down one X or the other. And if you shut down the X carrying the orange, okay, then you're going to be black. If you shut down the one carrying black, then you're going to be orange in that spot. So you get a blotchy cat. Okay. Now, males are only orange or black. They can't be calico because they only get one X. Okay. Now they can be. I actually, when I was in graduate school, I had a friend. Went over their house and they showed me there's a calico cat there, and I looked at it and said, yeah, it's a calico cat. And they said, well, pick it up, look at it. And it was a real friendly cat, so I picked it up and I flipped it over and I looked and it was a male. Whoa, okay, it's a male cat, not supposed to be calico. It turns out it was a Klinefelter's cat, something we'll talk about when we talk about chromosomes. It was a male, but it was XXY, so it had two X's and could do that. And it does occur. It's not uncommon for Klinefelters to be around. But for our purposes in this problem, we're going to have males either going to be orange or black, and females can be orange, black, or calico. Okay. All right. So let's get back to the problem. So here's the setup. Okay. We start out and we're going to do a 3-generation problem. We're going to start out with parentals, produce an F1, and they're going to produce an F2. All right. So the original parentals are an orange female. It's bred to a white male, produces an F1 litter. That F1 litter is all white. Okay, both males and females are all white. And then we take some of those males and some of those females, we cross them together, and we make an F1. Okay, excuse me, an F2. And here's the F2. We end up with 6 white males, 5 white females, 1 orange male, 1 tortoiseshell female, 1 orange female. Okay, all right. It's a classic top-down problem, right? It's just a problem here, except now it's also a bottom-up because we know the outcomes, and those are the fun ones and the easy ones because we've done the experiment, we know the outcomes. All right. So what's going to go on here? Well, questions are, you got these white males and white females in the F1. What's their genotype? Well, how do we do those types of problems where we got an unknown up here? Well, we look at the progeny and we just fill it back in, and that's all we're going to do. Okay, so as we always do, start out with your initial knowledge. Just write down what's given to you. What's given to us? We got a white male and a white female. Okay, so just write that down. A white male, a white female. How do you get to be white? Well, let's go back up here. A dominant light switch allele makes you white, absence of color. So these white females have to have that, and the males have to have it as well. All right. Now let's turn that around and look at their progeny. These guys up here are their progeny. What do we see? We see some oranges up here, right? It's orange males and orange females. Well, the females, because it's sex linked, are the more helpful ones at this point. Okay, so let's figure out what these are. Okay, well, in order to have color at all, it says that you have to be little w. So these orange males, these tortoiseshell females, these orange females, they all have to have recessive condition, right? Okay, all right. Well, where did they get that? If you produce some kids and they've got color and it takes 2 little W's to get color, where do you get it? Well, you have to get it from the parents, right? So over here somewhere, we know these have to be hets. It just basically tells us absolutely they have to be hets because in order to produce that by Mendel, right? Just fiddling around here with Mendel, okay, with this het times het cross down here, that's what you'll get, okay? So that's where they come from. We don't worry about the ratios. I mean, it's 6 to 5 to 1 to 1. I don't know what the ratio that is. We don't care. Okay. What we're doing is solving it the other way. All right. Now what do we know? Well, we've got this male here, right, down here. So he gets a Y, and now look what we're at. We only need 3 alleles now. I mean, we've almost solved it, right? We just got to figure out what these 3 missing blanks are, if we can do that. Well, what'll help us? Well, here's one. Here's an orange female. She's got her color turned on, and she's orange. This says to be orange up here, you got to have 2 orange alleles. Well, where did those come from? Well, just like the little W's, one must have come from this white female, and one must have come from this male down here, right? That's how you get them. Dad gives you one, gives the other. All right. Well, now we know exactly what Dad looked like. Okay, he was white, but really he was a carrier for orange, and of course he was a carrier for Y because he's a male. All right, now all we got to do is figure out this one allele right here. What'll help us with that? Well, what'll help us with that is this tortoiseshell up here, right? Okay, why? Because to be tortoiseshell, you have to be a het. Well, there's a B. There's a black allele. All right, where did that black allele come from? Well, it wasn't one spot, which is right here. So that female must really be a het. She would, if she had been allowed to have color, this F1 female, she would have been a tortoiseshell. Okay. All right. Now we can just kind of fill the rest of the stuff in. Okay. She's a tortoiseshell female. She has to have that. These are orange males. They look like that. And we don't know what these white ones are. All we know is they're white. We know it's a male. Okay. And this white female, well, because of this, she could kind of be anything. She could be orange. She could be she couldn't be black, right? Because there's no way to get 2 black alleles up to a female, but she could be orange or she could be calico. Okay, so we've done our best to kind of solve for those up there. Okay, and if you really want to, you could put for this female right here, she has to have an O, she has to have an orange right there. Okay, all right, well, that's pretty simple. That's great. We figured out this genotype. We didn't worry about ratios. We didn't do anything else. Okay, now here's a question. There's a color and a sex missing here. If this is the cross, what should we have seen up here that we don't see? Think about it for a second. Okay. Hmm. Something seems to be missing. Well, this black right there, an X, and that Y, if they had been put together and got color, we probably should have seen a black male, right? Right? Well, maybe one of these up here is a black male. It just got blocked out by color, but we should have seen a black male. Why not? Well, it's not very many progeny. Remember, fruit flies and stuff and things get thousands of progeny. You would see it there, but a mammal, I mean, it's a pretty big litter right here. So, you just didn't, by chance, didn't happen to see it. Okay. All right. So let's continue on and see where we are. Okay. Those can, what you can put there and work that out. All right. Now, the next part of the question is, all right, let's go back to this orange original parental, this orange female and this white male. Well, write down what you know. I mean, you ought to be able to write the orange female down without even thinking about it. She's got color and she's orange, right? So that one's easy. Okay, she's an orange female. She's allowed to have color. The males is the more difficult one. Okay, now we can solve two alleles immediately, right? We know that he's got to have a big W and he's got to have a Y, right? All right. What about these two in the middle right here? Why did I pick a B and why did I pick a big W? Well, because all the progeny turned out to be white, right? We'd put a little W there. We'd expect some of the progeny to have had some color, and they didn't. So our best guess is that he was a homozygous, and most parentals, remember, we do Mendelian crosses, they tend to be like that. Look like that, okay? And you had to get a B down here. Well, she didn't have a B to give, this orange female, so you had to do it that way, all right? All right. Excellent. Simple. I mean, it looked terrible at first, but it was really straightforward. Okay. All right. Now, we're still going to use Mendel. Let's think about it. Here's a rare tropical plant. Normal color is red. However, there are some pure breeding lines. Now, that pure breeding is really important, right? When you see that, it tells you that you've got some homozygous stuff going on here. There are variations that are found. Okay. So what are some of those? Well, apparently there's an orange and a yellow. These are parentals, so they're true breeding. When you cross them, you get an orange. When you cross those orange, you get a nice 3-to-1. Okay, that's pretty simple. We've seen that 1,000,000 times. Okay, you take the red and you cross it with an orange, you get a red, and you get a 3-to-1. Looks Mendelian to me, right? Looks like maybe orange is dominant over yellow, red's dominant over orange, and then we say, okay, well, what about a red times yellow? Because we got red and we got yellows. We get red again. And we get a nice little 3-to-1. Okay, looks like we've got a pretty straightforward system here. Wait a minute though, we've got three colors, right? We've got red, yellow, and orange. But we've got a nice little Mendelian system here. So what is this? It's one of those little simple allelic series, right? And it's a simple one. It's a dominant series. Apparently, red is dominant over orange. Okay, we see that right here. Okay. Orange is dominant over yellow. We see that from this first cross. And so red's dominant over both of them, right? Simple little dominant series. Not complicated, anything like, blood loci or something, ABO, where you got a codominant and incomplete dominant and all this stuff going on. You've got a simpler system. You got just a dominant series. Okay. All right. Well, you go, you raising these plants, you really like them. You go to a plant show, okay, and sure enough, there's another color there. Turns out that there's a white one that people have. And so you go home and you cross your red one, your pre-breeding red with this pure breeding white, you get red. And now you get a 3-to-1 of red to white. You go, wow, okay, that's interesting. So now it looks like I've got a third allele of white. So where does it go? Does it go right here as, like red, white, orange, yellow, or does it go red, orange, white, or is it the most recessive? Maybe it goes red, orange, yellow, white. Who knows, right? How do you go figure it out? You're a geneticist. You make a cross. You start crossing the white with the yellows and the white with the oranges and all that. So let's do that, okay? All right, so what do we got here? Let's cross it. We cross a yellow with the white. We just grab our yellow, pure breeding white. We get red. Oh, wait, that's not possible. Red's dominant over yellow, and that's weird. Well, then we go ahead and do it though. We cross these reds together, and now we get a 9, 3 to 1. So our red shows, is there, our yellow's there, and our white shows back up. Well, we keep doing it. We've got some other colors, so we say, well, let's try an orange times a white, see what we get. Whoa, this is really getting weird. We got a red here as our F1s. That wasn't even part of this, and it's supposed to be dominant. Yet we cross them again, we get a 9, 3, 4 ratio over here. Okay, everything's back, but it's in this 9, 3, 4. That's just really strange, okay? And then finally we cross a red and a white, and we get red. Well, good. Finally, that's telling us something. We've got a nice 9, 3, 4 over here. Okay, I'm really confused because all of these crosses up here tell me that red is dominant to everything, and they're perfectly Mendelian. Then I get down here and I got this weirdness. Well, 9, 3, 4, that's a ratio you should know, right? Okay, let's just think about it. What have we got? We've got a 9, 3, 4 ratio. That's an epistatic ratio, one of the ones you have to know, like 12, 3, 1 or 15, 1 or whatever. Okay, how do we get 9, 3, 4? 9 plus 3 plus 4 adds up to 16. 16 comes from one cross, a dihybrid het times a dihybrid het. You can use any alleles you want over here, but you're going to have two loci, an A and a B locus, and they're both going to have to be heterozygous. That's how you get 16 parts, right? All right, and I said when you do that, you can write some stuff down. Think about it, okay? Grab one of the alleles and make it look like this. In fact, we did. And what we end up with is that we've got a light switch, just like with the kitty cats, okay? This time, if you have two little B's, you're white, you turn off color, so it's not a dominant, it's the other direction. And this time, color is the dominant one. So getting one big B will allow you to have color. Then what you got to do is look at these other alleles and figure out what they're doing. Well, that's that little dominant series, okay? Red, orange, and yellow. You don't have to add in white up here because white is being produced by this interaction, this epistasis between these loci, all right? So let's think about what do we do now? We've got this yellow times white, write it down, okay? Produces red, and those two reds cross together to make your F2 over here, produces a 9, 3, 4, all right? That's producing this. So what does it tell us? It tells us that we had to have color, and we had to be hets. We had to be dihybrid, right? Okay, we get some yellows down here. Yellow is the most recessive of all of them, so they must be two little Y's to get that, so that must give you that, okay? Red comes from having some red alleles right there, 9, okay? Yeah. Yep, so how do we get them? Well, remember we said they were true breeding or pure breeding, so this is a pure breeding yellow back up here. It's allowed to have color, and it's yellow. This is a white, but we needed to get some red alleles sent down here, so it must be this, and then that makes it white, okay? Now we've got all of this together, okay? We've got 3 alleles, and they're hets, but it doesn't matter. We get a 9 by having a red, and we don't care what the other allele is. Could be another red, or it could be a yellow, okay? And we end up with color over here. Then we get some yellow ones with color. Then we get white, just turns everything off, right? Okay, 9, 3, 4. All right. How about this second one? Well, orange times white gives me red. Same story, same data, same piece of information. Orange times white, okay, gives me red. Cross two reds, we get a 9, 3, 4. This time the orange is there instead of yellow. So that must mean there's some orange alleles hidden up in these reds. Okay, well, there surely is. They came from this orange up here. So it's the same argument, okay? We need some reds, we need some orange, we need some color, we need some lack of color. Crossing those together, that's the same as this up here. Yes, well, this is an orange individual that's true breeding. We end up with this. This is a white one, but we need to know that it's hiding the red allele, so it's there, okay? And we get a 9, 3, 4. Wow, okay, that seemed horrible at first, but simple Mendelian, and we just have to deal with the fact that we've got 3 alleles. All right. Now, those are pretty simple. Let's torque it one more level, okay? Let's just make it a little more complicated, but guess what? It's not going to be any more difficult to solve. All right, here's some cute little mice. This guy up here is known as a Goodie. It's a gray mouse. That's the thing you see. It's a European house mouse. The thing you see running around your house, and especially in the winter, they come in and run around. People scream when they see them, but they're the same. You can go to the pet store, or you can go to Scientific Supply, and you can pick up a nice yellow one. It looks like this, or you can get these very common ones, as you see, are white mice. Excuse me. And these white mice are just the same species, same guy. They're just more inbred or different than the others. Okay. All right. So what do we got? Oh boy, here's another one of those paragraphs. All right. A locus in mice is involved in pigment production. Obviously, it's pigment because you got color. When parents heterozygous at this locus are mated, 3/4 of the progeny are colored and 1/4 are albino. Sounds like a simple dominant recessive to me, right? Colored versus albino. Albino is white. Okay, that's an albino right there. Okay. The other locus controls what color you have. All right, so this first one is an on-off switch, right? 3-to-1 on-off. So that means color's dominant over lack of color. Then once it's lights on, you have to figure out what you got. Well, when two yellow mice are mated, you get a 2/3 of the progeny are yellow and 1/3 are Goodie. Okay, so let's just write that down. Let's think about it a minute. What are the modes of inheritance here? Well, a 3-to-1 albino, that's simple Mendelian, right? Just kind of, we can write big A's or big B's, whatever we want. That's simple Mendelian. So color is dominant over absence of color or albino, right? Now we got 2-to-1. Where's 2-to-1 ratio come from, right? 2-to-1. 2-to-1 comes from having the hets, the 2's always associated with the hets, and the other one, the Goodie, is associated with a homozygote. Now, we don't care whether it's big Y's or little y's. It doesn't matter. Okay, I'm just putting them like this, but it doesn't matter. What it matters is that when you get either 2 little y's or 2 big y's, as I've drawn here, they die. All right, it's a recessive lethal. So what is the mode of inheritance? Well, it's a slightly complicated. It's epistasis with a recessive lethal. Oh my goodness. All right, right? This is recessive lethal over here. That's recessive. Recessive meaning that you have to have 2 of them, right? It kills you, so it's a lethal. But it interacts with this one over here to actually give you these colors up here, right? So it's epistatic with a recessive lethal. That would be the proper way to define the mode of inheritance involved here. Holy cow. Okay, that's all right. Let's see, let's try it. Let's think about that and work with it though. When yellow mice are crossed with albino, they produce an F1 consisting of 1/2 albino, 1/3 yellow, and 1/6 Goodie. Okay, if that's true, what are the probable genotypes of these yellow mice times a Goodie? So always write down the given. I'm crossing a yellow times an albino, right? And I'm going to need 4 alleles because I got epistatic, 2 loci. And this is what I'm producing, a 3-to-2-to-1 ratio, right? Okay, so let's think about that. 3-to-1. Do the given first, all right? First question you're probably asking is, wait a minute, I see a 1/2 and a 1/3 and a 6. Why'd you go 3-to-1? Well, because it's simple, you know, that's grade school math there. We're just going to take 1/2, 1/3, 1/6. When you have ratios like that, fractions, you always try to get them in a common denominator, which is 6, and then those can cancel out, and we come up with a 3-to-1. We're better off, us, working with ratios, right? So 3-to-1 ratio is what we've got, right? That'll work for us. So that's all I did. Okay. 1/2 of 6 is 3, you know, 1/3 of 6 is 2, etc. All right. So now what can we do? Well, this is the yellow one up here, so she's got to have that. This is an albino over here, this mouse, so it's got to have 2 little a's. This has got to have 1 big A. All right, we got 3 of the alleles figured out already. Okay, this one's albino. Okay, right? That's albino progeny. All right, so one of those A's came from here. Where did the other little A come from? Yeah, okay. Same old stuff we've been doing since day one, right? Had to come from here. So this yellow has to be a het, right? This albino is a homozygote because that's the only way it can be albino. And that's for the first locus. Now we're going to just figure out what the second locus is. Okay. All right. Well, we know this one's yellow up here. Okay. All right. And we know these guys have color, so they're picking that up from here. Okay. All right. They're going to be like that. They're going to be hets. They got to be hets because this one's given little A's over here. We do yellow. All right. All right. Okay. Now she's yellow. Remember that if you're big Y, you die. So to be yellow, you got to be big Y, little y, right? That's definite there. These have color. These have color. Okay, they're going to pick up some of that, right? All right, so we get an agouti right here. Okay. We get this a Goodie down here, okay? And then we get this yellow up here. We've got that, okay? So we know this one, but we don't know this one over here. All right. So here's where it's slightly complicated. We've got to use now this ratio to try to figure out what's going on. We can't know what's over here, right? We can say, well, I'm pretty sure there's a little y. Okay, I got that, all right? Because I know I got two little y's here. One came from there, one came from there. But where did we get this? You know, I don't know what this is. So if we want to be complete, we have to try it out. So how do you do it? Throw a couple together. Say, okay, I know this side of the equation. I know most of this side. Let's just fill in what could be. Okay, if it could be that, follow a little Mendelian process along, and you see, I don't think that's going to work. Okay, right? Because that's going to be a one of those, and one of those, and one of those, and only one of those. And that's going to not look right, okay? What it's going to take is, well, let's go back and try this, okay? That's going to give me one, that's one of two, one of one, one of one, one of two, one of one. Get all this. If you look at that, what have we got, all right? We've got 2 plus 1 of these, okay? So that's going to be yellow. That one's going to be a Goodie. But look at over this 2 plus 1 over here. These are recessive. Whites are albinos. Albinos, doesn't matter what you got over here. So in order to get a 3 to 2 to 1 ratio, it had to be this cross, not this cross. And so look what we did. We used logic, we used ratios, we used Mendel, took everything to do that. And when we do that, we realized that this albino, even though it's albino, had to have been an albino yellow. Okay. If it had allowed to have color, it would be yellow, not a Goodie. Okay. All right. That's kind of fun. Now, let's try one more part of that. Remember the same thing. If yellow F1 mice are crossed among themselves, what are the phenotypic we expect in the progeny? What proportion of the yellow progeny would you expect to be true breeding? In other words, breed true. Okay. Now remember with epistasis, that can be a little weird. So just think about it. Now normally breed true means homozygous, but when we go to epistasis, sometimes that's not always true. You can still get things that are red, always producing red, even though they're heads, okay? So let's take a yellow, okay? Allowed to have color, color's yellow, allowed to have color, yellow, it's F1, so we know they're heads, okay? Simple Mendelian, okay? Pull up this silly chart one more time, last time, I'll show it to you. It's one of those crosses, okay? Just the het times het cross, nothing different, okay? Just happens to be a little weird, okay? We take my little method that I like to use Punnett square if you want, I don't care, but big A times big Y, little y's, 2 of those, and 4, and 2, and fill it all up, okay? And then we just figure out what these are, okay? Well, these have color, these have color, these have color, these have color, these are a Goodie. Excuse me, these are albinos, right, down here, okay? All right, so those are albinos, right? Now these have color. What color do they have? Well, there's some a Goodie, and then these guys are yellow. We get that, so these are yellows. And then these two are Goodie. And so our final ratio actually is 1 albino, okay, there's 3 of them, to 2 yellows, because there's 6 yellows, to 3 a Goodie, so a 1 to 2 to 1 is my final phenotypic assortment ratio, okay? Now the second question is, what proportion of the yellow progeny will breed true, okay? What proportion of the yellow progeny will breed true, right? Well, these guys are not going to breed true because if you take that, you're going to get some albinos, right? No matter what this does over here. Okay. What about those over there? Okay. Right. What proportion of the yellow progeny are going to breed true? Okay. True breeding. Well, I don't think any of the yellows are. Okay. Why not put these little stars by here? Because those are the only ones that are actually going to breed true. These will breed true. These albinos, because this will always make it albino. It doesn't matter what's over here. Now, it's not breeding true in a genetic sense, as far as a genotype. It's breeding true in a phonetic sense, in the way the phenotype looks. Right? So if you take these albinos and you cross them among themselves, they'll always be albino progeny, even though genetically they're not pure. Okay, these will be pure. These are true breeders. Okay, and then these Goodies up here will do that. There's none of the yellows that will actually do it. Okay. All right. Okay, well, that worked through some really difficult problems, but you see, they're not that hard. Okay, you have to pull them apart, think about them, and work them. At first, they're terrible, but then Mendel's going to do most of the work for you. You just let Mendel work it out. You can figure it out from there.